So let me introduce today's speaker. Today's speaker is Travis. He started his, I guess, his master's in Toronto, and he did his PhD in Bielefeld. And now he is back in Canada as a faculty at Dalhousie University. I know Travis for a long time. I think I've met him at CPM or some such venue for the first time, and he likes to travel a lot. So if you want to invite Travis, he will certainly come, and if you let him know well in advance, then he will even learn a local language before actually coming. So he's great. So invite him everywhere. Okay, so he is going to talk about computational pangenomics with the R index, and with that I pass the word to Travis.
Thank you for the introduction. Thank you for inviting me. Hello, everybody. Hi from Canada. This is going to be a talk about pangenomics with the R index and friends. So the friends is going to be sort of the R index and derivatives, but also a bunch of stuff sort of related to which options, alternatives to the R index, also just friends, as in this is work with a lot of people over about 20 years. So I'll get started because there's a lot to go through. So the goals of this talk, what am I actually going to try to say and tell you about? Well, I was planning to start this by telling you all about the FM index and how the FM index works and why the FM index is important, why pangenomics is important, why it's important that we have data structures like the FM index that do things like the FM index, but that work with genomes instead of that work with pangenomes, so genomic databases instead of single reference sequences. However, if you go to the PANGAIA page, that's sort of all in there. So probably everyone listening knows these things. At least I hope they know most of these things, because I'm going to skip this, because that will let me talk about more stuff. So instead of sort of all that background, I want to argue that we need a pangenomic version of the FM index, and it should have three properties. So it should probably have a lot of other properties as well, but the three I'm going to focus on today are that it should be, oh, I have to start the timer. Okay. Calm down. Okay, it should be elegant, which means that it should be theoretically nice. There should be sort of meat to it. There should be like interesting insights. And we should be able to prove nice bounds and really talk formally about what we're doing. And this keeps the theorists happy, and that's nice because they're about half our community, or at least they're a big chunk of our community. And it's sort of a waste if they can't be involved. And they are useful because they do provide insight into what we're doing. If we don't have theoretical elegance, we're sort of flying blind. It should also be practical, because that keeps the other half of our community happy. And it means that we're actually doing something useful. So we want good implementations. And we want it to work on large data sets. We want it to be scalable, all this kind of thing. And the last thing is it should be versatile. Because if we just focus on one problem, which usually we focus on, it should find occurrences of P and T and O of length of P plus awk time, or something polylogarithmic at times, something polylogarithmic in N maybe. That may not be what people actually want. So it should be versatile, it should solve lots of problems, it should be adaptable. And I'm going to talk about whether our current solutions have these properties, and why and why not. Okay, so the FM index has these properties. I hope you're going to believe me. It's elegant. It appeared in Fox and the JACM, and its derivatives appeared in like Fox and Stock and the JACM. It's practical. It powers Bowtie and BWA and a load of other tools in bioinformatics. So if you're doing something with bioinformatics, usually you start with sequencing, and the sequencing usually that happens with software that's built on top of an FM index. So please excuse me for this whimsical term of phrase. It's versatile. It goes forwards and backwards. It goes up and down. It goes round and round. And actually, it goes forwards and backwards. It's bidirectional. It really does go forward and backwards. The up and down, there's this thing called the alternating BWT. It goes round and round. There are circular BWTs and FM indexes for finding circular patterns. It finds common substrings, it finds rare substrings, it finds maximal exact matches and minimal unique matches and parameterized matches and order preserving matches and all this kind of stuff. So you can do a lot of things with the FM index. And that's what we're aiming for. And it's not that, it's a hard act to follow. So let's keep these three things in mind. Okay, so some background to what we got stuck on and sort of what we've tried before we got to the R index. Okay, so Veli Makinen and Gonzalo Navarro, they were already working on a run length compressed FM index by the Dymax workshop, the Burroughs-Wheeler transform 10 years later. Now, 2 years ago, we had the 25th anniversary of the FM index. So that workshop was the 10 years later was in 2004. So with Yoni and Nico, they developed into the RLCSA and applied it to indexing genomic databases. So the nice theorem from that work, which it appeared in several papers with updated versions. So sort of summing it up, we can store a text T of length n in O of R space, where R is the number of runs in the BWT of T, such that given a pattern P of length m, in O of m log n time, we can find the BWT interval containing the characters, I should say preceding, not precedings, occurrences of P and T. I hope everyone, the BWT is the permutation of the characters in T, where they're sorted by the suffixes that follow them. I hope people sort of know that, because otherwise this is going to be hard to follow. It takes like a talk to explain what the FM index is, so I'm just going to skip that. Okay, so finding the BWT interval for P tells us the number awk of P's occurrences in T's, but not their location. So it tells us the number because each character in the interval precedes an occurrence of P, which means the number of characters in the intervals is the number of occurrences of P. So with a normal FM index, what we can do is store the suffix array entries that are multiples of a value G. And then with the BWT, you can backward step. So you can go from a character in the BWT to the character that precedes it in T. So that's a backward step. So from each cell in the BWT interval for P, we can backward step until we hit a character whose position is one of these sampled suffix rate positions. Okay, so that takes, for each of the characters in the interval, this is going to take something like O of G time, times log n or something like that. And it's going to take, log n bits for each of the n over G sampled positions. And so this is going to, if we choose G to be a log to the 1 plus epsilon n, then the SA sample takes about n over log to the epsilon n, which is in little o of n bits. And locating each occurrence takes log 1 plus epsilon. That's actually a var epsilon. It's not the same epsilon. You'll see it's written a little bit differently. It takes about logarithmic time. But the problem is, so with a normal, if you're indexing one genome, that's great because something slightly sublinear is not really going to affect your size. And logarithmic, you're willing to pay that. But if you're indexing a genomic database, then R, the number of runs and the space you need for the run length compressed Borel Wheeler transform, it can be thousands of times smaller than n. And it generally, it is. It's about, you divide the space, you divide by the number of genomes it is, because humans are nearly genetically identical. So in this case, n over log c epsilon n is huge compared to the rest of your index. So, but if you want to reduce the space by increasing the gap between sampled suffix array positions, then you're going to slow things down proportional to the decrease in the space. So the product of these two things, the product of the space and the query time, is going to be at least linear. So this means the RLCSA, if you want it to be small, it's going to be slow to locate. And if you want it to be fast to locate, it's going to be huge. So this is sort of a problem with going from indexing single references to indexing genomic databases. Much of the versatility of the FM index, it's derived from these n over polylog n space auxiliary data structures. So sampled RMQs over the LCP, and people who know and love this stuff will know what I'm talking about. Other people, just trust me on this. So for example, the SA sample, normally it's n over polylog n, and you get polylog n query time. And to work with a pangenomic reference, these have to be redesigned or they have to be completely abandoned if we're going to use our polylog n space, because they're just too big. Little o of n is no longer good enough. This is the rules have changed now. We're in a different world. So for a long time, it seemed that the RLCSA, it couldn't be practical, it couldn't be small and fast and versatile at the same time, able to do all these things the FM index does. Okay, so with the R index, it seemed like we'd hit this wall, so it was a dead end. So Veli et al. Started pursuing other approaches to indexing genomic databases. So I'm going to talk about the ones that I've sort of been involved in. Actually, also been involved in CDOGs a bit, although I never really understood how they worked. But I have been involved in LZ and SLP-based indexes, hybrid indexes. I'm not involved in founder sequences, but I was sort of down the hall from lots of people who were, and variation graphs, which I'm not really involved in, but I dabbled in sometimes. So as I've written here, there are lots of other things, CDOGs, if people listening might know Bloom filter tries, things like that. But I hope that just going through this stuff, I mean, that would take a whole nother talk. So just going through these, I'm going to try to give a flavor. So the LZ and the SLP-based indexes. So the ideas behind these are basically the same, and they go back to the mid-90s, to Juha and Esko's paper. Back then, it was the workshop on string processing, now it's Spire. And you can also build these things based on string attractors, which is sort of the latest thing that people have done. So let's just do it for the LZ77. If you partition T into its LZ77 phrases, then the first occurrence of every substring of T, by the definition of the LZ77 parse, it has to cross a phrase boundary. And if you know the positions of all of the occurrences of P that cross the phrase boundaries, which are called primary occurrences, then you can deduce the positions of all of the occurrences that are completely contained within phrases. Right? Because everything is completely contained within a phrase, it has to have been copied from wherever that phrase was copied. If you go back far enough, you have to hit one that crosses a phrase boundary. So it's the same thing with the SLPs. Then you say, well, everything at some point, it has to be contained within the expansion of a non-terminal, crossing the boundary between the expansion of that non-terminal's sort of left child and the expansion of the non-terminal's right child. Hideo calls this kushi or something. It's like it gets skewered. Okay. So you can store the Z polylog n space data structure. Z is the size of the parse. And with, so for I from 1 to M in polylog n time per occurrence, you can find the colexic, so you have this tree, maybe a Patricia tree or a Z-class tree or something, above a grid, and you have a tree beside the grid. You use these to find the colexicographic range of the phrases ending with P1 to I, and the lexicographic range of the suffixes of T, starting at phrase boundaries that start with Pi plus 1 to M, and then you sort of look in you take these two ranges, you take their cross product, and you look in that range, and if it's got something in there, then you've found a phrase boundary which is preceded by P1 to I and followed by Pi plus 1 to M, and so you found a primary occurrence. So these are elegant. Okay, so there's lots of nice stuff in there. You can like shave off log factors and get lots of data structures involved in. It's really fun working with these. Potentially they're practical. Like they're quite competitive. They're fairly small. They're fairly fast. But it's really hard to do anything with them except exact pattern matching. So Gonzalo has fought like crazy, and I think he can now do document listing. I'm not sure if he can do document counting. But it's not like an FM index where, just things, other kinds of queries just drop from the sky and you can do whatever you want. Getting an LZ-based index to do something other than exact pattern matching is fairly painful. So it's not versatile, at least not yet. Okay, speeding up. Hybrid indexes. These basically what you do, you take the LZ77 parser, something like that, and you say, okay, I'm not interested in patterns more than length M. So you just take the stuff that's within distance M of a phrase boundary, and you concatenate these substrings. This is your kernel string, and you FM index that. So it's like a mix between a hybrid between an FM index and an LZ index. You can find the primary occurrences quickly. And then you can do the same stuff that you did, the same data structures that were with LZ indexes to find the secondary occurrences. The thing is, it's practical fairly for short patterns. It's versatile because basically everything you could do with the FM index, you can sort of do with a hybrid index. It's not elegant. Okay, and I say that as somebody who helped dream these things up. They're just the bound on the pattern length is annoying, and it's just the theory is kind of ugly. Okay, founder sequences. The founder sequences for a genomic database are a relatively small set of artificial genomes. So the idea is, okay, some founders arrived on an island, and then generations later, everybody on the island, their genomes can be obtained, could have been obtained by recombination events from the founders' genomes. Actually, I was just reading a few days ago that Quebec is actually an example of this. They say that now there were relatively few people who actually came over and settled in Quebec, and that millions of people are descended from these like 2,600 people. Okay, founder block graphs are similar, but they're graphs instead of sets of sequences, and the sequences in the database, they can be represented as paths through the graph. The thing is with founder sequences, like hybrid indexes, you're sort of taking a compressed representation, a lossily compressed representation. The kernel string in the hybrid index doesn't represent everything. I mean, you can't, just from the kernel sequence, you can't get back the database. And just from the founder sequences, you can't get back the database. You need the recombination events. So yes, you can get it back, but you have to do post-processing. Your index itself is not indexing your database. So the index can return incorrect results. For example, P can occur in the founder sequences, but not in the database itself. So you've done these recombination, you've constructed these founder sequences, and they're not in the database itself. So you have patterns in there that aren't in the database. So a match then can be a chimeric match. So though we can correct the results afterwards, its potential errors may mean it's less elegant and versatile. Okay, variation graphs. According to the page, the PANGAIA page, everybody here loves variation graphs. I sort of like variation graphs too. They're like, I don't know if you need to hear this, but they're like automata accepting the language of genomes, possibly modified such that you can index them easily so that you can blow them up and embed them in the brain graphs. But again, variation graphs are lossily compressed representations of the genomic database. So the index can return chimeric matches. So there are lots of elegant results about variation graphs, and there are practical results about variation graphs, but they don't seem to appear in the same papers. So it's not clear that there's sort of an agreed implementation that is theoretically elegant and practical. The theoretically elegant stuff doesn't seem to get implemented. Or the stuff that gets implemented at least is not written for the theorists. I don't know if that's fair. You can correct me afterwards if there's time. So Wheeler graphs were sort of an attempt at a common language that the practitioners could love and the theorists could love and talk to each other. But I don't know if that's really caught on. If it's actually being used for this conversation. Okay, so let's get to the R index. How much? I have to go faster. Okay, so the R index really, apart from the RLBWT, the RLCSA, which started like decades ago, the R index really got started at DC616 when Polykriti and Pretza took the first step towards beating the linear time-space trade-off for assay sampling with the RLCSA. Remember, you had this thing where if you made the gap bigger, it got slower, and if you made the gap smaller, if you made the gap between the sample positions in the assay bigger, then you slowed it down. Okay, so with O of R space and M log n time, we can locate one occurrence, just one, assuming that P does exist in T. Okay, so this, we're going to call it the toehold, and we're going to use it to locate all of the occurrences. So I actually came up with this name. I'm not sure that that's to my credit. We're going to call this the toehold lemma by Polykriti and Pretza. So we can store T in O of R space such that given the pattern P in M log n time, we can locate one occurrence of P in T if it exists. And I'm sorry, this I used Beamer, the pause thing, not pause, but only. And so the proof shifts up as I reveal more and more. So what we're going to do, we're going to store the suffix array entries at the run boundaries in the BWT. We perform a backward search for P such that at any point I, after I backward steps, we know some QI, which is going to be a position in the BWT interval, and we know SAQI, so we know that suffix array entry. We know the position in T of that character in the BWT. And it's a match for, so T SAQI to SAQI plus I minus 1. So it's that character comes, that character in the BWT, BWT QI precedes a match to PM minus I plus 1 to M. So the last I characters of P. Okay. So we've got the entry and we've got the interval and we've got the position of one character. So we can do that when we start because the empty suffix sort of matches everything. So we can always choose Q0 such that it's a run boundary. The BWT Q0 is at a run boundary. Okay. So for I greater than 0, so we have the interval and we have BWT QI and we have its suffix array entry. If BWT QI matches PM minus I plus 1, which is, so if the BWT character matches the preceding, the next character we're going to look at in P, which is the preceding character, Then QI, we just do an LF step to get QI from QI minus 1. And the position in T is just the SA QI minus 1. We just move left in T as well. So that's easy. But suppose the interesting case, if BWT QI minus 1 doesn't equal the character in P, then the last occurrence of the character in P before BWT QI minus 1, or the next occurrence, the last occurrence is the end of a run, and the next occurrence is the beginning of a run. If neither of those are in the interval, then P doesn't occur in T. If one of those, at least one of those in the interval, is in the interval, then we can choose it as our new candidate. Okay, so we can we can reset QI minus 1 to that position, to jumping up or down, and then we reset QI to that, and then we have a match and we can go on as before. So, okay. So that was the toehold lemma, and Nicola told me about this on a hike after DCC 2017. And we had this long drive back to Salt Lake City, and by the end of it, we sort of had the basics of this stuff. So with the RL BWT, you can find the BWT interval for P. With the toehold lemma, you can get one suffix array entry. So in that interval, so you get the position at one occurrence. So if you could support phi and phi minus 1. Where phi, given a suffix array entry, phi of that position in T, it gives you the preceding suffix array entry. And phi minus 1 is phi inverse, just if you give it a suffix array entry, it gives you the next suffix array entry. So if you can find the interval and you can get one entry, suffix array entry in the interval, and if you had phi and phi minus 1, then you could just list everything else in the interval. So you could get all of the suffix arrays, so all of the positions of the matches of the occurrences of P. So it turns out we figured out you can do that using predecessors. So the observation is if BWTI, if a character in the BWT is equal to the preceding character, then phi of SAI, what you can do, those two characters, they're the same character in the BWT, so an LF step is going to map them to also neighboring characters. So phi of SAI is going to be, you could map them and then take the phi and then add 1, and it's the same as taking phi, right? So if you take the LF step and take phi and then add 1, it's just like taking phi. Okay. So if you iterate this, if you say, okay, if these two characters are beside each other, if these two characters are beside each other are in a run in the BWT, and if you do the LF step, then those two characters are still are still within a run. And you do a bunch of steps. You do up to D steps, and they're always within the same run. Then you can say, well, phi of SAI is the same as phi of SA LF to the D, so doing D LF steps of I, and then plus D. So to sort of undo the effect, you do D LF steps, then you do phi, and then you add D to undo the LF steps. So if D is the smallest non-negative value, such that BWT LF to the D of I is not equal, so you do D LF steps, and then you're not within a run. So until then, you've always been in a run after every LF step. You're not in a run. Then the T of SAI minus D minus 1 is the last character in T at or before the character you started with, that is the first character in a run in the BWT. Okay? And so you follow this line of reasoning. If you store an O of R space data, a predecessor data structure with the positions in T of the first characters in the runs in the BWT with the corresponding phi values as satellite depth. So the phi values at the position, at the characters in T, which are at the beginnings of runs in the BWT, then you can answer phi queries in log n time. And symmetrically, you can answer phi inverse. So this is a bit much for one slide, but if you've been working on this stuff for like 15 years, then this is super simple. And don't just take my word for it, take the word of Jamal Balazougi, who when I wrote to him, I emailed him this, and he wrote back, and he said, that is cute result and so simple that one wonders why it was not discovered before. So at least for Jamal, this is a really easy slide. So from this, you get the R index. So I'm sort of splitting the R index into two, and this part I'm calling the R index, and the other part I'm going to call the RCST. So we can store T in O of R space such that given a pattern P in M plus off log long time, we can locate all the occurrences of P and T. I think this is elegant, in all modesty, if I say it myself. It's reasonably practical, at least since we developed this technique called prefix reparsing that you can actually use to build big BWTs for pangenomic data sets. But without any of the auxiliary little O of N space data structure, the FM index, it's not versatile. So PFP seems practical and versatile. It's not super elegant. There are more elegant competitors, but I'm not sure they're practical. So I just to note. Okay, so why do, this is what convinced me that things need to be versatile, because a bunch of us, Alan Quinlay, Teher Moon, Christina Boucher, me, Ben Langmead, and Giovanni, we collaborated on this carefully engineered, thoroughly tested implementation of the R index. It got into Recom 19, it got into the special issue of JCV. We had as tools, we had two papers in the special issue, one of which was tools, which just said how you install it, how you run it, everything. But it only supports exact pattern matching, so it can't be really used to do many things. So it doesn't actually get cited a lot, unfortunately. So exact pattern matching, people think that you use exact pattern matching to do approximate pattern matching. And traditionally, yes, you did this thing where given the pattern where you want to allow cares, you cut it into k+1 pieces, you do exact pattern matching, you do seed and extend. The problem, that works for short and highly accurate reads and a single reference genome. But for long and less accurate reads or a multi-genome reference, if you take a long read that's got like 10% error rate and you cut it, so K is going to be like, if it's 5,000 characters long and you're allowing a 10% error rate, you're going to cut it into 500 pieces of length 10. That's not going to be useful. You're going to get too many matches. The same thing if you cut a pattern into little pieces and then you match in a pangenomic reference, well, each little piece could match thousands of times. So you actually don't want to do exact pattern matching, you want MEMS. So can we make it versatile? Maybe. So, well, sort of, yes. Nicola and Dominic, they defined the string attractor. So sort of working to add functionality to the R index, I think that's what got Nicola started on string attractors. So the string attractor of a string to be a set of positions such that every substring has an occurrence containing a character in one of those positions. So it's like the LZ77 parses a string attractor. BWT and T have string attractors of size O of R, as do differentially compressed suffix array, inverse suffix array, and the LCP. For example, if BWT I to K is contained in one run, then you can think about it. The LCP of LFJ For j greater than I and less than or equal to k, it's just going to be LCP of j plus 1. So if you take the differentially compressed LCP array, then you can take a run and sort of it has to equal. If you LF everything, it's going to equal that. Okay. So it follows that you can build from results by Gonzalo and Nicola and Tomek and lots of people. It follows that you can build straight line programs, so grammars of size OR polylog n that provide log n time access to BWT, T, suffix rate, inverse suffix rate, LCP. And if you augment these grammars, then you get polylog n time RMQ range minimum query, previous smaller value and next smaller value queries over the LCP. Therefore, you can build an OR polylog n space compressed suffix tree with O polylog n time. If you want to get it down to like R log n and log n time queries, that's going to take some work, more than one slide. So this is elegant and versatile because suffix trees make so many things easy, but this is not practical. I don't think anybody's going to implement this. Maybe on a dare. So, okay, so we wanted to see what can we get practical in O of R space and or essentially, and that's actually practical. So Hideo, Tomohiro, and I realized we can use the toehold lemma to compute matching statistics. You also need random access to T for part of this. And thus you can find these maximal exact matches. Okay, so this was in a paper in CPM 18, I think. And then it was in the special issue for Danny Breslauer. So suppose we know QI, so this is toehold lemma again. We know QI and SAQI and the long end, that's the beginning of a longest match. It doesn't have to match the whole suffix of P, but it's a longest match, starts there. Okay. So if the BWT character matches the preceding character in the pattern, great, we just do an LF step and the length gets one longer and we're fine. If it doesn't match, then the preceding occurrence. Now, if you had the interval for the suffix, then maybe you're not going to have the character you want in the pattern in that interval. But it means if you're looking for a longest match, you're going to be jumping either to the preceding one or to the succeeding one. Okay, because these are lexicographically ordered. So the longest match, the suffix that follows the BWT character, it has to be that's preceded by the character you want. It has to be either following the preceding occurrence of that character in the BWT or the next one. So you either jump up or you jump down. And those are the beginnings and ends of the run. So it's just the toehold lemma again. So you can pre-compute a threshold. So these are the beginning, the end of a run of a certain character and the beginning of the next run of a certain character. You can compute a threshold between those, such that if you're above the threshold, you jump up, and if you're below the threshold, you jump down. You don't have to store anything else, just this threshold. So Max Rossi noticed that this threshold is actually, you can choose it to be the position of a minimum in the LCP between these two runs of the same character. And that makes it easy to build. We didn't notice that for CPM 18, and that's why we didn't actually build the thing. So once we know, so once you know where you have the pointers in the matching statistics, then you can do another pass. You do this pass from right to left, you get the pointers in the matching statistics, and then you do a pass from left to right, and you use random access to T, which you get with a grammar. And this takes, you compare only a linear number of characters because if you get a long match to one of the characters, then you know that to one suffix of P, then you know that the next suffix of P, you get a match that's at least that length minus 1. So you don't have to do all those comparisons. Okay. So from this, we built Money, which is Finnish for multi. It also happens to be the name of a friend of mine, nickname. It just needs the RLBWT, the assay samples, and the run boundaries, and a straight line program for random access to T. It doesn't need support for phi and phi minus 1. It doesn't need the beautiful predecessor data structures that we came up with on that drive, unless you want to list more than one occurrence of each now. So I think Money is fairly elegant. It seems practical. It'll appear in Recom 21, and finding MEMS is a step towards being versatile. Although needing 2 passes over P is a bit frustrating, because you can't do streaming in real time. So while we were writing Money, we noticed that if we use the grammar, instead of doing random access, we use it to these longest common extension queries as well as a random access, then actually you only do need this 1 pass over P. And we don't need to store the thresholds at all. That is, when we're going along and the BWT character doesn't match, then we do an LCE query between that position, which we know where we are in the text, and the end of the preceding run of the character you want. That's at a run boundary, so we have the suffix array entry. So we have its position in T, and then the next run of the character we want, that's run boundary. So we have its position, so we can do these LCE queries, and whichever one is longer, we get the length of the match, and we can go to the one that's the beginning of the longer match. So originally we called this funny for phony money, because it seemed like a hack, because we're using a grammar to get LCE queries. We didn't think we could get a good bound for that. But then we realized if you use a locally consistent grammar, then the queries take logarithmic time, so everything runs in m log n time, so it's actually not so phony. So it is actually fairly elegant. It's practical, it's versatile. And just before we submitted it, Christina realized that you can use it with nanopore sequencing, because since it's online, it's streaming. You can be watching the matches and deciding whether or not you think the string, the base calls from the nanopore, whether you think this is actually what you're looking for. And maybe if it's not, you spit the DNA strand out of the nanopore. Apparently you can do this. So Ben said, "Oh, that's great." And he contacted, because Mike Schatz at Johns Hopkins as well, he'd just done this with Sam Kovaka and some other people. He'd done this software called Uncalled for spitting out DNA strands you don't want. And so we started optimizing Phony to recognize boring DNA strands you don't want to keep going with, and to spit them out of a nanopore. So we perform way more backward steps than you do jumps. So most of the time, the BWT character matches the character in the pattern. So you just do the LF step, which is fast. But the LCE queries were taking most of the time. So Ben and his student Omar suggested just using the thresholds instead of the LCE queries. So doing it streaming, doing Phony, but with the thresholds instead of the LCE queries. And we don't know the lengths of the matches, and we never know the lengths of the matches. So we just count the number of LF steps, which is a lower bound on the length of the match. And so this is called the pseudo matching length. And you use this as your statistic instead of the real matching lengths. That's horrible. Okay. Theoretically, as a theorist, I would never think of this because my mind would block such a hideous idea from occurring to it. But it's also extremely fast, it's compact. You don't need the suffix array samples, even at the run boundaries. You don't need the SLP. So it's really tiny. Also, this is the part that really offends me. The pseudo matching lengths actually worked better in the experiments than using the real matching lengths, which is just terrible. So using a pangenomic reference also works better. Just to know, we did try it with a single reference, and the pangenomic reference works noticeably better. And also, using the whole sequences, the whole pangenomic reference. We tried it with the founder sequences, and that doesn't work as well. So we called the software Spumoni for streaming pseudomoni, and it'll appear at recomsec. And then I remembered that sputare means to spit in Italian. So it's like Moni who spits. But Moni, my friend, does not spit. She would be offended if I suggested she did. Okay. So summary, so working in OVAR space and no longer being able to add little O of n auxiliary data structures seem like, it feels like when you start doing this, it feels like moving into a house that has low doorways. So my brother is 2 meters tall. So he has problems in Europe because he keeps trying to stand up straight and whacking his head on stuff. So if you're when you move in somewhere and there's this, for the first few days, you keep bashing your head on the door frame, but then you learn and you just start automatically ducking. So we have to learn that we don't have those auxiliary data structures anymore. What can we do? We don't have them unless we add, we blow things up by a logarithmic factor. We get the RCST, and then we can do whatever we want. It just won't be practical. So I think we're moving towards having something that's comparable to the FM index. It's elegant, it's practical, it's versatile, and it works for pangenomics. So I should probably, so the summary, the FM index is it's elegant, it's practical, it's versatile. The RLCSA, it's elegant, but if it's practical, it's not versatile. If it's versatile, it's not practical. LZ-SLP, it's elegant, it's practical, it's not versatile. Hybrid, not elegant, hideous, disgusting. Maybe it's practical, it's versatile. Founders, maybe. Variation graphs, it's elegant. No, sorry, I was saying if it's elegant, it's not practical, and vice versa. It is versatile. I'm not sure, I could be convinced that it's all three. The R index, yes, yes, no. RST, yes, no, yes. Money, yes, yes. Maybe it's versatile. Funny, maybe it's elegant. It's practical, maybe it's versatile. Spumoni is definitely not elegant. It is practical and maybe it's versatile. Now, Tomasz, should I go through the 6905 projects and the open problem, or should I let people ask questions?
I think you can go ahead.
Okay, so in the semester that just ended, I ran a class at DAL, and DAL let me do it even though only one DAL student showed up. So I'm just wanting to sort of boast about the students in this course. It was so fun, so much fun. Okay, and I'm probably going to mispronounce the names. So I didn't have much time in this talk to talk about the prefix reparsing or Wheeler graphs, and I didn't speak about tunneling at all, but I have these high hopes for combining them. So Uwe Bayer proposed tunneling, so this idea of sort of squishing, merging paths in a Wheeler graph. And he showed it can shrink the RLBWT by up to a logarithmic factor. In theory, it does actually pretty well in practice. So tunneling optimally is hard. Tunneling the BWT of a massive data set, even suboptimally, isn't easy. But Andre Balázs, Adrián Goga, and Lucas Panzani Ramos, so at Comenius and Campinas, they're studying how you can use prefix reparsing as a pre-processing step to speed up Uwe's tunneling algorithms, and without worsening compression much. Davide and Marco, so the prefix reparsing, it can break a substring into two blocks, two phrases, A and B, even if A is always followed by B and B is always preceded by A. That's a waste, because that should really be one phrase. So it's wasteful. So Davide Genzato and Marco Oliva are experimenting with a repair-like algorithm to optimize the prefix reparsing as a post-processing step to discover to discard phrase breaks when discarding the phrase breaks reduces the overall size of the dictionary in the parse. So far, it seems to work. And last night when I was writing this up, I thought if it keeps working, I should get them to try, because PFP was inspired by rsync and spam sum and other algorithms that use this stuff called context-triggered piecewise hashing. I should go back and get them to try it with rsync and spam sum. FM indexes suffer from a lack of locality. Joni Siren, he proposed breaking the BWT up into blocks, storing the ranks at the beginning of the blocks because we have a small alphabet, so that doesn't hurt much. And then you can rearrange the blocks. So you have some table that gives you sort of a summary of where the blocks are. You rearrange the blocks so that a backward step from one block tends to take us to a block stored nearby in this rearranged block rearrangement. So Joni did this for VG, but it's buried in VG. So now Jamie Deng, who at Tsinghua with Wenkai Han, is implementing a version that we should be able to use with Moni, Phony, and Spumoni. Which is good because otherwise, how are you going to speed up Spumoni? Because it's really just doing LF mapping. So I said that this is something I did talk about. LZ and SLP indexes are not yet versatile. I put the yet in there because Pavel Yakov and we had a paper at CCCG 13 that showed that you can use these for finding the longest substring of P that occurs in T quickly. And that was improved by Aben et al. At CPM 18. So for his class project, Yunnan Gao, who's actually the one student at Dalhousie, figured out how to use LZ 77 indexes or things like that to compute the matching statistics in the MEMS. And this, the space is in terms of delta and gamma and delta, where gamma is the size of the small string attractor and delta is this even better measure of repetitiveness. And so you can use these to get MEMS and matching statistics. So Sara and Giuseppe, when we're doing this jumping up and down, we're always jumping. Remember, we're always jumping to a run boundary. So can we speed up the LCE queries by bookmarking the characters at the run boundaries in the parse tree, so that to get to them, you don't have to descend all the way from the root. So from the LZ77 index stuff, we have this bookmark trick for grammars. You don't have to descend for the current candidate because you can keep a finger on it, and as you LF step, you just move the finger to the left in the parse tree. Also, this is something really cool. They've shown that Phony can quickly compute minimal unique matches as well as maximal exact matches, so mums. So that's going to be nice maybe for finding informative substrings that tell you sort of which individual could this match. If it only matches one, then you, okay. And so I should ask Yunnan if he can also do mums. So the open problem is with a normal FM index and a little O(n) space auxiliary data structures, you can find the MEMS of P with respect to T, and you can get their BWT intervals. So you know how often they occur, how often each mem occurs. With O of R space, we can't really do that. We can get the MEMS, but we don't get the intervals. So we don't know how often the MEMS occur. So the mums, we're telling if the interval is just one character, but we want to know really the length. So you can get the LCP arrays from phi and phi minus 1 if you augment this. So you can get the intervals by listing the contents, but that could be really slow. You can backwards search for each mem separately, but that could take quadratic time, m squared time in the worst case. This is actually more or less what BWA mem is doing. In the worst case, BWA mem is quadratic in M. It works with a single reference because you tend not to get big overlaps of mems. But with a pangenomic reference, you probably will. And of course, with R log n space, you can use an RCST, but that's impractical. So is there a better way to do that? I would be really happy if somebody could figure this out. And now that's my question. And sorry, we have like 2 minutes left, but now I give it to other people to ask questions or complain about how I've shafted their favorite data structure. Sorry.
Okay, so I have a question regarding the nanopore application. So basically, you are saying that, okay, I can analyze these strings in a streaming fashion and find the matches, and if I find enough matches, then I know that the string is boring or whatever, right?
Yeah.
But the problem is that sort of the nanopore data, I mean, it doesn't come as a streaming. It does come in chunks. So basically, you cannot base call a single base. You have to base call a chunk. So you know, it comes in a blocks of, let's say, 50 bases or so. And so does that change anything in the?
Not really, because I mean, you don't have to run it real time. So actually, you could do money instead of funny. It doesn't. And so I thought the blocks were actually longer than that. I thought the blocks were like a few.
No, it depends.
Okay, so I thought it was a few hundred. Because the nanopore, the strand doesn't go through the nanopore all that quickly. So it turns out I mean, it's what? Each pore is giving you
450 bases per second.
Yeah, it's not that much, right? So it might take us a couple of seconds to recognize that the pore is sequencing something we're not interested in. But that's still
okay. So there is also edit, you know, but there are like 500 pores sequencing in like at the same time. So.
Oh yeah. So that's the thing. It's the slowness, the few seconds to realize that the pore is not sequencing something we want. That's not because the processing time is so high. We can actually process fast enough to keep up with 500, or if you get like the shoebox full of min ions, what's it called?
Green ion.
Yeah, then we can probably even keep up with that. The thing is, the base caller, the error rate is fairly high. So you don't want to like kick the strand out just because you go for a section and you think, yeah, I'm not getting a long match. The thing, if you look over several seconds and you say, how long are the matches? Well, they're going to get broken up. But if you're looking for matches, if you've indexed a pangenome of the species you're interested in, so if you're sequencing the DNA from that species, even with the errors, over a few seconds, the lengths are going to tend to be significantly longer than if you're sequencing another species which is unrelated. Because the errors are going to happen in both cases, but the lengths for when it's a matching species, the lengths are going to sort of be the lengths between errors. Whereas if you're sequencing a DNA that's from something else, it's going to be, well, the errors are going to break it up, but also you're just not going to get as long matches. Probably I should refer this question to Ben or Christina or Omar, but I can send you the Recom Sec paper. Also, Mike Schatz is on this. So actually, one thing you might be interested in, because I know you do the nanopore stuff, and I'm over time, so I'm not offended if people disappear. But one thing we haven't done yet is try to hook the index back into the base caller, because at the moment, what you could do, your index, it's actually not that hard for the index to tell you. For the index to tell the base caller, if the next character you tell me is an A, then the longest match with the database is of this length. If it's of length, if the next character you tell me is a C, it's of this length. So it might be that the base caller is saying, I don't know if it's an A or a C, but maybe there's a little bit more evidence that it's an A. So I'm going to say A. But if you actually looked at the BWT and you looked at the data set, then you'd be saying, yeah, well, an A doesn't match anything, but a C is what I expect to see. So at the moment, the information is just flowing from the base caller to the index. So it might be interesting to also have the index telling the base caller sort of what it expects to see.
Okay, that makes sense actually. So, because there is a something called this is a neural network, and there is usually something called CTC layer there, which is basically a probabilistic distribution over bases instead of a real string, and then like everything gets collapsed, and that's the final string. So that might be an interesting thing to match against.
Well, I see there's this I saw a paper about something called SquiggleNet, which is doing something like this. So
that's one step even further. So
okay,
so you can also try to match the electrical signals, but that's even funnier.
Yeah, so I was suggesting that we could try to index the electrical signals, and that idea went over like a lead balloon.